All right, I'm going to talk through the acid-base balance lecture. Hopefully it helps. If it doesn't, I'm sorry, but I hope it makes it a little bit more easy to understand. This is how I understand things as I talk through things. All right, so the normal pH ranges from 7.35 to 7.45. Optimal pH is 7.35 to 7.45. pH, it's important to note that pH, so just think pH, there's an H on pH. So you're measuring the amount of hydrogen ions. The more hydrogen ions you have, the lower your pH. So it's an inverse relationship. The lower your pH, the more acidic you are, the higher your pH, meaning the less amount of hydrogen ions you have, the more alkalotic you are. Acid enters the body from a lot of different places, and it always needs to be excreted or reabsorbed to maintain the balance in a sweet spot of 7.35 to 7.45. Acids are produced in our body as a product of cellular metabolism on a daily basis. So that just happens. Carbonic acid is a part of the byproduct of carbonic acid, but the end result of aerobic metabolism is carbon dioxide. Lactic acid is a byproduct of anaerobic metabolism. So the breakdown of glucose, which is glycolysis, it ends with lactic acid. Sulfuric acid is the oxidation of sulfur containing amino acids. And then phosphoric acid results from the metabolism of phosphoproteins and ribonucleotides, which are used as an energy source. And the last one is that can be acids in the body are ketone bodies, which are fatty acids that are broken down via ketogenesis, which we learned about earlier. All right, so this formula is the important formula of the buffer system and how it works between the lungs and the kidneys. So anytime you're on the CO2, carbon dioxide side, just know that that is the lungs working, and then the bicarb side, that's the kidneys working. So an easy way to kind of remember which way things shift here. So think back to pH. pH is low, so you're acidic, which means that you have a lot of hydrogen atoms in your body. So if you have a lot of hydrogen atoms in your body, you're going to want to get rid of them or buffer them. And bicarb is the buffer. So by buffering it, all buffers do is help maintain your pH by either combining with a hydrogen or getting rid of it. Or creating a hydrogen, I should say. So either creating a hydrogen or getting rid of a hydrogen, that's buffering the pH. So if you have a lot of hydrogen, bicarb is going to combine with it and make carbonic acid. I'll write that. Carbonic acid. Now, with the help of the enzyme carbonic anhydrase, carbonic acid will break down into carbon dioxide and water, and then carbon dioxide is exhaled through the lungs. So that's how you get rid of this hydrogen, is that you move to the left. So if you have a lot of hydrogen, you have this process wants to happen a lot and quickly. That's why you breathe quickly, because you're going through this process quickly and a lot to get rid of all that extra hydrogen. The opposite is true. If you do not have a lot of hydrogen, then you're going to want to retain CO2. And so think respiratory depression. You're going to try to keep a lot of this so you can go through this process a lot. So if you have a lot of carbon dioxide, you're able to combine with water and use carbonic anhydrase to make carbonic acid. And then since carbonic acid is a really weak acid, it just breaks off into a hydrogen and a bicarb. So then you get your hydrogen. So I know you see that we're making both a hydrogen and a bicarb. So you would think like the ratio is not going to change, but that doesn't matter. pH is only measuring the amount of hydrogen. It doesn't matter if you're adding hydrogen and bicarb. You're adding hydrogen, so you're going to decrease the pH. Just important to remember that anytime you're going from here to here or here to here, you have to use carbonic anhydrase. This is a weak acid, so it just breaks off. So the control mechanisms of the acid-base balance use the chemical buffer system, the kidneys and the lungs. The chemical buffer system uses bicarb, phosphate, plasma proteins, and hemoglobin. This is how it uses bicarb, phosphate, plasma proteins, and hemoglobin. The kidney's role in the control of the acid-base balance is 3 parts and includes the reabsorption of bicarb, the excretion of hydrogen, and the excretion of hydrogen as ammonium. So bicarbonate here is the most important extracellular buffer. It's the first line of defense against the alterations of the acid-base balance. So if your pH is off, this is what's going to happen first. And that's why your lungs react so quickly, because it's going through this. Phosphates can be either inorganic or organic. Inorganic phosphate is an extracellular buffer. The chemical HPO4, so buffering, same thing. So just think this is buffering. So by the phosphate HPO4 -2, when it gets buffered, aka add or subtract a hydrogen, it goes to H2PO4. And so that gets rid of the free hydrogen, so that affects the pH. So that's how phosphate does that. But that's an inorganic phosphate. Organic phosphates are all of the adenosine phosphate groups. So think adenosine triphosphate, ATP, adenosine diphosphate, ADP, and adenosine monophosphate, which is AMP. So ATP, ADP, AMP, glucose 1-phosphate, and then 2,3-diphosphoglycerate. Plasma proteins also act as extracellular buffers. So when you think plasma, I think of what's in your plasma: albumin. Albumin is the main plasma protein, and it has a negative charge. Therefore, it has a role in buffering hydrogen, because hydrogen is positive. Albumin also has a role in binding with calcium. So albumin binds about 40% of free calcium. So in acidotic states, there is a lot of hydrogen atoms floating around, which makes albumin really busy to bind to the hydrogen ions. And since albumin is so busy binding to hydrogen, there's not a lot of albumin left to bind to calcium, which means that there's a lot of free calcium floating around. The opposite is true. In alkalitic states, there is not a lot of hydrogen atoms floating around, so the albumin's looking for something to do, so it binds with all the calcium that's out there, which gives you a decrease in the free calcium. So hemoglobin is an important intracellular buffer, and it plays a pivotal role in the acid-base balance. So this is a vessel here, and here's my little red blood cells with hemoglobin carrying oxygen, and then hemoglobin drops off oxygen, and then with cellular metabolism, it creates carbon dioxide, and then it goes back and it goes to the lungs, right? So this is like the capillary situation happening here. So when carbon dioxide comes back into the vessel, it attaches, it combines with a water molecule, and to do that, remember, you have to use carbonic anhydrase here. And so in your vessels, you now have a hydrogen and a bicarb, okay? So you don't want free hydrogen just hanging out in your vessels because you would be super acidotic. So what happens is hemoglobin attaches to the hydrogen and it kind of gives it a ride up to the lungs. And then this reaction happens. So when the red blood cell and the hemoglobin reaches the lungs, the process reverses, and then carbon dioxide and water are formed. So hemoglobin detaches from the hydrogen, and then so you're left with this, and then it creates carbonic acid, and then with carbonic anhydrase, it goes to CO2 and water, and then you breathe off the carbon dioxide. So basically, you just need to remember that hemoglobin hydrogen hitches a ride with hemoglobin up to the lungs. So it's not just a free floating hydrogen, okay? So obviously the lungs act first, and then if that doesn't compensate, then in about 24 hours, the kidneys will kick in, okay? 3 roles of the kidneys. The first role is the absorption of filtered bicarb. So filtered bicarb, just know that everything in the lumen here, this is after the glomeruli, so anything that's coming through the lumen is filtered by the glomeruli. So this bicarb here is technically filtered because it's gone through the glomeruli. So here's your proximal tubule. This is happening shortly after the glomeruli filters. So this is your proximal lumen. And here's your cell that's in between the blood vessels and then the lumen. So what happens is you have bicarb here. You're going to use a hydrogen to combine with the bicarb, which is going to go to carbonic acid, which with carbonic anhydrase, I just wrote Ca, that is going to break down into carbon dioxide and water. The carbon dioxide and water is going to diffuse into the cell. The carbon dioxide and the water is going to go back to carbonic anhydrase, to go back to carbonic acid with the help of carbonic anhydrase, which gives you a bicarb and a hydrogen. And so this hydrogen just gets recycled, and this process goes over and over and over. And then the bicarb that it's creating by going through this kind of circle here comes back out into the blood. And so that is what is reabsorbed, because this was filtered and now we're taking it back. So it's filtered and then it comes through here and it comes back into the blood, aka reabsorbed bicarb. So then you also have, this also will connect some other dots, because through your lumen here, you have this transport mechanism that kicks out a hydrogen and brings in a sodium. And you'll remember that sodium is reabsorbed in the proximal tubule. That's because it's exchanged for hydrogen here, and then it comes out with bicarb, or it uses the ATP sodium potassium pump and comes out there. Chloride also comes in through the blood, and that's how that gets excreted. Okay, so big takeaways are the filtered bicarb comes down, breaks down all the way to carbon dioxide and water, comes into the cell, goes through the opposite process, and then the bicarb is kicked out. Okay. Oh yeah, this was in bold. The site of action for the carbonic anhydrase inhibitor happens right here. So if you use the medication whatever that, I'm really bad at pronouncing medication names, but the medication that inhibits carbonic anhydrase, this medication blocks this from happening, and so it can't, that can't get moved into bicarb and water, so this part of the equation can't happen. So then you can't reabsorb bicarb. What happens is that you just excrete this process just keeps going down and becomes urine, okay? Okay. This part says when the extracellular fluid volume is expanded, it inhibits the reabsorption of bicarb in the proximal tubule. So when your fluid volume is up, this doesn't happen as much. When your fluid volume is down, it increases the reabsorption of your bicarb. A volume deficit, if you remember, also stimulates the renin-angiotensin system, which stimulates this exchanger here, the sodium-hydrogen exchanger. So this is going to be working a lot more. So a lot more sodium is going to be going in, and a lot more hydrogen atoms are going to be coming out. So since you have a low volume, since all of this sodium's coming in, it's going to come out through here with bicarb. Or it's going to come out through the sodium-potassium pump. And so it's going to increase the bicarb reabsorption because it's going to come out with sodium. And since the renin-angiotensin system is it happens because of a decrease in volume, water follows sodium, and that's why you get increased blood volume and increased volume of sodium and water when you use the renin-angiotensin system. Treatment of this is volume replacement. So if this is happening too much, volume replacement will fix that problem. Renal excretion of hydrogen atoms by a titratable acid. Did I do that? Okay, I did. Yeah, all right. Titratable acid is one that can lose protons in its reaction. Inorganic phosphates assist with the excretion of hydrogen. So 15% of phosphates that are not reabsorbed, so 15% of phosphates that don't end up over here, are obviously in the lumen, and those are available to interact with hydrogen in the tubules. The renal excretion of hydrogen as a titratable acid occurs mostly in the distal tubules and the collecting ducts. So distal tubules and the collecting ducts. So remember, the phosphates, the titratable acids happen in the distal tubules, but what we just talked about before was happening in the proximal tubules. The cells of the distal tubule collecting ducts contain 2 active transport systems, which uses ATP because they're active and they need energy. The hydrogen pump and the hydrogen-potassium pump. And just like we talked about in a different lecture, potassium and hydrogen move in opposite directions, and then this one just takes energy to kick hydrogen out. They move hydrogen into the lumen and move potassium in. So when you have a hydrogen in the lumen, it attaches to a monohydrogen phosphate, so one hydrogen and a phosphate group. When these combine, you get H2PO4, which is dihydrogen phosphate, and then that can be excreted as urine. So for every hydrogen excreted, there is 1 bicarb reabsorbed in the blood, and that is because you're going through this, but it's like the same thing with the bicarb reabsorption, but you're not creating bicarb over here to be excreted like you were over here, because this is using phosphate and not carbonic acid. So the only bicarb that you're getting is from the inside, which you're getting carbon dioxide and water from the blood and creating this over here, so that you're getting bicarb over here. So that's why you only get 1 bicarb over there. Excretion of hydrogen as ammonium. Renal excretion of hydrogen as ammonium occurs in the proximal tubule. The ascending loop of Henle and the collecting ducts. So it happens in 3 places. Proximal tubule, ascending loop of Henle, and then the collecting ducts. The proximal tubule cells produce NH4. Okay, so the proximal tubule cell, so this is a proximal tubule cell, they produce NH4. Inside these cells is glutamine, which we get from the blood. So the glutamine comes in, and then the glutamine is broken down with the enzyme glutaminase to glutamate, plus NH4. So you get glutamate and NH4. Oops. I wasn't supposed to write that. So, yeah. Yes, so you get glutamate and NH4. You also have NH3 in here. Glutamate is then metabolized by alpha ketoglutarate, and then, so I'm just going to do alpha keto G, and that goes to CO2 and water. And then eventually this goes with carbonic anhydrase, it goes to carbonic acid, and then a hydrogen and a bicarb, which that gets reabsorbed into the blood, same way that it did before. And then your extra H comes out here, NH3 goes to NH4, goes to the urine. So let's just read through this. Glutamate is then metabolized by alpha ketoglutarate and then into carbon dioxide and water, and then goes all the way down the chemical reaction to bicarb, which is then reabsorbed in the blood. NH4 exists in equal amounts to NH3, so you got NH3 and NH4 inside the cell. NH3 is a lipid soluble and easily diffuses into the lumen. So that just easily just floats on out there into the lumen. NH4 is not lipid soluble and is transported by the sodium hydrogen exchanger. So you got a hydrogen and a sodium. So what happens is the NH4 will break off and become NH3. That extra hydrogen will come out and exchange with sodium, and then your NH3 will come out into the lumen. And then out here, it combines with a hydrogen that you kicked out to create NH4 to go to the urine. At this point, some NH4, which is ammonium, is excreted in the urine. The remaining, because remember this is happening in the proximal, the remaining ammonium is reabsorbed in the ascending loop of Henle. And then the ascending loop of Henle, it is reabsorbed and placed into the interstitial fluid of the kidney by substituting potassium by substituting it for potassium on the sodium potassium chloride transporter. So instead of potassium hopping onto that co-transporter, it switches spots with the potassium, and NH4 takes the spot of potassium. But if you have a lot of potassium in your body, aka hyperkalemia, potassium competes for that, and ammonium is not able to switch places with potassium because you got too much potassium, and then it won't transport, and then it won't excrete. Okay. The concentration of ammonia, NH3, is much higher in the interstitium of the kidneys than in the collecting ducts in the lumen. NH3 in the lumen binds with hydrogen and forms NH4. NH4 cannot be reabsorbed and is excreted as urine. the excretion of NH4 increases the urine pH. So that is why you see, not only is there hydrogen ions, but you have ammonium. That's why the pH of urine is usually like 6. All right, that's the end of this one. I hope it helped me just talking through it. So even if you don't listen to it, help me. Thanks.